everybody, it's Mr. Matthew here for From Molecules to Organisms, video number 3 in our video series. And in this video, we're going to talk about really homeostasis and feedback mechanisms. What we're going to do is we're going to talk about how either individual cells or whole organisms get information and respond to that information. And we're going to do so by talking about feedback mechanisms, both positive and negative, and some examples of each. We're also going to talk about how cells respond to different external signals and how they will either use energy or not use energy to move things in or out. And then we'll also talk about some of the active structures within cells and how those help maintain homeostasis. So here we go. So before we get started, let's define the term homeostasis. So when we use the word homeostasis, what we're using this term to mean is we are maintaining a relatively stable state. What that means is if we have a condition in the body, let's say human body temperature, and so we want to maintain a relatively stable body temperature of 37 degrees Celsius. And as a result, when we break down our food and we produce some heat as a byproduct, we use that to maintain that body temperature. But what happens if the temperature gets hotter than that? We go outside, it's excessively warm, one of those warm days, and so we're getting this external heat plus the breakdown of food. And so now what we're going to do is we're going to have our nerve cells and our skin cells are going to communicate that information to our brain that, wow, it's really warm. And then the temperature regulation that takes place in our body is going to send signals out and cause you to release sweat through sweat glands. And so what this means is that we're going to respond to this to try to get our body back to that 37 degrees Celsius. We start with the stimulus, we have some sensors, we then go to control, and then as a result, we have some sort of output or effector in this case that is really important. So what we're seeing here is in feedback system, this is when a system changes in response to the products created by the system. So in this instance, when you sweat, you're going to basically create a layer of liquid on your skin that is going to evaporate. That's going to be evaporative cooling, and hopefully that's going to lower your body temperature. When your body temperature comes back down, it will turn off this message that it's too hot, and you won't be sending the signal to cause yourself to do additional sweat. This is a form of negative feedback. I'll show you some other examples of negative feedback, but this is one example of negative feedback. So let's talk about positive feedback. And when we talk about positive feedback, that's when the products of the system actually perpetuate the system. They cause the next thing in the system to take place. And so the example that we're going to use here is injured tissues releasing chemicals in the blood that activate platelets to cause blood clotting. So let's say you have a wound and a wound takes place and we get a cut in a blood vessel. And what will happen is that this is going to proceed to create a large cascade of signals that the damaged tissue is going to give off some chemical signals. Those damaged chemical signals are then going to recruit things like platelets, which we have here, and it's also going to cause the release of certain proteins. And what we see here is that each one of these steps in this blood clotting process is actually going to turn on the next steps in the process. So the products that are made, such as the production of fibrin or the recruitment of platelets to this area, are going to release signals that don't shut off blood clotting, but are actually going to turn on the next steps in the blood clotting factors. So we refer to this as a positive feedback system. The other common positive feedback system we sometimes refer to is with childbirth. And what happens with childbirth is you will have uterine contractions, and those uterine contractions send out a signal that causes an increase in intensity of uterine contractions. And that's why uterine contractions get much more close together and more intense until the baby is born. The products of the system, which are the contractions themselves, perpetuate more contractions, and so that's a feedback. So we're doing this in a very simple way, and I would like you to keep that in mind, but I do want to give you a sense of, I'm going to pull back the curtain a little bit and give you a sense of what's involved. When I look at blood clotting, this is not just one step or two steps. It is actually a whole bunch of different steps. And so, we've got platelets, and those platelets release what are called tissue factors, and those tissue factors go through an initiation phase, and then there's an amplification phase, and you can see all of these different proteins, and you don't need to know any of these. But what I want you guys to remember is that the products of the system, whatever is made, so let's say we make the platelets come together. When the platelets come together, they're going to do something that causes the next step of the clotting process to continue, and that's what makes this a positive feedback system. So Let's talk now about negative feedback. And so in negative feedback, it's the case when the products of the system turn off the system. And regulating blood sugar is another example of a homeostatic goal of our body. So in our body, what we try to do is we try to maintain a relatively constant level of blood sugar. That way, if we eat food and we take in more glucose into our bloodstream, what we'll do is we'll rapidly store this. We don't want our blood glucose levels to go too high. And similarly, if you've gone too long between meals, let's say you've had to, rush to the bus because you overslept, and now it's been 18 hours since you had dinner before and you haven't had lunch yet. Now what you could do is you can release stored glucose in your body in order to maintain a certain level of your glucose so that you have a constant supply of energy for the cells throughout your body. So let's talk about this. And I'm going to start at the top of this, and I'm going to talk about how glucose levels rise. And so in this instance, what's happened is you've taken in a meal. As a result of that meal, you've broken down those foods, you've created glucose through all the enzymes we talked about in the previous video. And now what's going to happen is the pancreas is going to recognize that we have more blood in the sugar, and it's going to release a chemical compound called insulin. Insulin is going to then cause certain target cells in your body, mostly in your liver, but also in your muscles, actually is going to uptake that glucose and store it in a form called glycogen. As a result of this process of the insulin causing cells to pull glucose out of the bloodstream, your blood glucose levels will fall down to a normal level. All right, so again, now we've gone too long between meals, and now as a result, the pancreas is going to release this other chemical called glucagon. And so glucagon is actually going to go to the liver cells, maybe go to the muscle cells, and it's going to cause you to break up the glycogen stores of glucose and release that and bring your glucose levels back up into a normal range so that all of your cells have available glucose. This is two things. One, this is negative feedback, the product of the system where, high blood glucose levels actually turns off the release of glucagon. Low blood glucose levels turn off the production of insulin. It's also an antagonistic system. There's two hormones that are working in order to bring you back into that homeostatic middle balanced state. One raises glucose levels, the other lowers it. And this is another feedback mechanism that we see, the antagonistic work of two different hormones coming together that your body will use and other multicellular organisms use two antagonistic systems to try to maintain homeostasis. All right, so now let's go from the whole organism down to the cellular level. And so what we're going to talk about here is what your body does in order to have passive transport and active transport to move materials across a cell membrane. So most obviously what we have in this case is we're going to talk about the movement of something like water across a membrane. And so in our first instance, what we have here is we're going to take a cell or a model cell, and in that model cell, we're going to have equal concentrations of solute, and in this case, we can envision maybe this is something like salt that doesn't move across a membrane very easily, and we have an equal concentration of salt and equal concentration of water, both inside and outside the cell. And as a result, we refer to this as an isotonic solution, and water moves back and forth very easily. There's no energy involved. Water's going to go really easily. But let's say we take that same cell and we drop it into a really salty solution. And so now there's more salt outside, and there's less water outside. And as a result, what we're going to see is the water is going to rush out of that cell and the cell is going to shrivel up. And we call this a hypertonic solution. The solution outside is going to cause the water to rush out of the cell, trying to get an equal balance of water and solute both inside and outside the cell. This causes the cell to shrivel. The last example is, well, what if we put that same cell in a higher concentration of water? Rather than a 20% solution like we did in situation A, we're going to put this in a 10% solution. And so now what we'll see is that the water is going to rush into the cell, and it's going to actually make it what's called hypotonic, or it's going to cause it to swell. And there's a limitation to how much a cell could swell. If I put this in a 100% water solution, maybe it's going to be so much water trying to rush in, it would cause the cell to burst or lyse, and as a result, the cell would be destroyed. In reality, the cell concentrations in most cells tend to be more like 4 or 5%, but there are many instances where you could put a cell into a solution and it would be too concentrated. Now, why am I talking about water and salt? Well, in this case, what you'll see is the water is just moving across. Water is a molecule that moves across cells very easily, and this is done by passive transport. The cell's not expending any energy to move this water from one side to the other, and therefore we refer to this as passive transport. Now, let's contrast that with some large or charged particles. Large and charged particles tend to need some help moving across the membrane. And furthermore, sometimes we want to take large or charged particles and we want to create disequilibrium. We want to have more of a particle on one side or more of a particle on the other. And so let's now look at where we have an area outside the cell and we have an area inside the cell. And we've got all of these sodiums on one side, and we want to move some of those sodiums across the membrane. This is actually something that happens in your cells, in nerve cells. In order to be able to send a nerve impulse, you will actually concentrate sodiums on one side of a cell in order for you to be able to have a nerve impulse go down. There's other ions involved, but under simplified cases, you want to have a lot of sodium on one side. So in this case, sodiums are both charged, and what you'll see is we'll have more of it on one side. So in this instance, in the way this diagram is set up, sodiums want to go from one side to the other. And so this is actually another form of passive transport. And this form of passive transport is actually a really easy one to model, in the sense that what we have is these charged particles. They are going to move from high concentration to low concentration. But the problem with them is that they can't get through these phospholipids very easily. So they need to get some facilitated transport to move through. So they need special protein channels to help them through this process. So now what we want to do is we want to see what happens when we want to reestablish this disequilibrium. Well, now what's going to happen is we're actually going to pump those sodiums from the inside to the outside. And in this case, what we want to do is we want to create a situation where there's more sodiums outside and more potassiums inside. So we're going to have to spend some energy. If you spend energy to move materials across a membrane, we refer to this as active transport. And in this instance, we're going to use a specific pump to pump these materials across. Again, why are we talking about movement of materials across a membrane? Well, this is actually how your body creates a situation of homeostasis. There are times when your cells are going to want to maintain a situation in order to create a stable state. In this instance, this allows cells to communicate using these sodiums and potassiums to send information down a nerve cell. Without the expenditure of the energy, this organism can't send a message from one part of the organism to another. And so in the earlier instance, when I talked about recognizing the room that was too warm, if that was the case, you would never be able to send the signal out from your brain to open up those sweat glands if you didn't invest this energy on the cellular level. So this is all tied back together. All right, so the last thing that we're going to talk about is how you can use specific organelles, in this case we're going to talk about the lysosome, in order to maintain homeostasis within the cell. And the reason for that is that the lysosome is this membrane-bound organelle. It's really, really tiny. It's blown up way too large in this particular model. And if we get a light microscope, you usually cannot see a lysosome under a normal light microscope. But what we find here is that inside the lysosome, there's all of these digestive enzymes. So let's say you have a situation where you need to break something down. You have an old organelle that is no longer functioning, and so what you want to do is you want to recycle the macromolecules that were used to build that and maybe build some new ones. What you would do is you would set up a structure where the lysosome would be able to go into a vacuole and break down and recycle those cell parts down into monomers using those digestive enzymes. This then makes those monomers available in order to build additional structures. And so not only do you have the ability on the large scale system, on the multicellular system, to send messages from one part of the body to the other, or to have an individual cell communicate with its external environment, even with inside cells. There are specialized structures that help maintain homeostasis within the cell, and the lysosome and its digestive enzymes are one of the examples for that. All right, so hopefully you could provide evidence that homeostasis maintains internal body conditions through a wide variety of feedback mechanisms and small-scale cellular processes. This would include the positive feedback that occurs during a wound and the formation of a blood clot, negative feedback using hormones such as insulin in order to maintain blood sugar levels. You also will see that cellular processes such as passive and active transport and the recycling of materials using the lysosomes help to maintain homeostasis. All right. Just so you know, the interactions at the molecular level, such as like how to produce insulin, is not expected to be on a state test, but we may go into some of those details in class. All right, I hope this was helpful, and I'll talk to everybody soon.